Ruchi Patidar. Today I am discussing about scope and method of biophysics. In this whole video, I will cover it in two parts. First is scope and method of biophysics, and second is amino acid. I will explain scope and method of biophysics. I will discuss in subtopics. First is introduction, second is biophysics, third is discoveries of biophysics, scope of biophysics, molecular and subcellular biophysics, and the mechanism of biophysics, biophysical methods, and biophysical techniques and application. Introduction. Biophysics is the branch of knowledge that applies the principle of physics and chemistry and method of mathematical analysis and computer modeling to understand how biological system works. Like that, mathematics plus physics plus biology plus chemistry. It is the modern interdisciplinary field of science leading of new approaches for our understanding of biological functions. Next is, what is biophysics? Biophysics is a specialized sub-area of biology. It is the science of physical principle of life itself and its biological process. Biophysics is an interdisciplinary science that explains the law of biological process. Biophysics speaks of all level of biological organization from molecular scale to whole organism. Discovery of biophysics. First is optical accept of the human eye. Second is first law of thermodynamics. Third is theory of hearing. Fourth is Brown's motion, and fifth is osmosis process. Sixth is non-equilibrium thermodynamics, and seventh is last discovery of X-ray, emergency of radiation biophysics. And next topic is scope of biophysics. What is scope of biophysics? Mainly is two type. First is biophysics topics based on relative size of the subject, and second is biophysical techniques and applications. Biophysical topics based on relative size of subject is three type. Molecular and subcellular biophysics, physiological and anatomical biophysics, and environmental biophysics. And second is biophysical techniques and application. This is a more topics. General biophysical topics, imaging biophysics, medical biophysics. Biophysics applies the power chemical physics, chemistry and maths in understanding health, preventing disease and inventing cures. Next is molecular and subcellular biophysics. First is structure and conformation of biological molecule, structure function relationship, comfortable transmission, ligand binding and intermolecular binding, diffusion and molecular transport, membrane biophysics, DNA and nucleic acid biophysics, amino acid biophysics, protein biophysics, energy flow and bioenergetics, thermodynamics and carbohydrates, nucleic acid, stratific mechanism, kinetics, molecular mechanism, DNA and RNA mechanisms, and allosterics. Next is mechanism of biophysics. First is biophysics is the study life at every level from atom and molecule to cells, organism and environment. Their work always aims to find out how biological system works. How do protein mechanism work? How do system to nerve cell communicate? How do protein pack DNA into viruses and RNA into viruses? How do viruses invade cells? How do plants harness sunlight to make food? Biophysical methods. Biophysical methods are technically to study the structure, property, dynamic or function of biomolecule at an atomic and molecular level. They encompasses a range of techniques including microscopic spectroscopy, electrophysiology, single molecule method and molecular modeling. The ideal biophysical method would have the capacity to observing atom level structure and dynamic of biological molecule in their physiological environment, in example like in vivo and in vitro. In would also permit visualization of the structure that form throughout the course of comfortable conformational changes or chemical reaction regardless of the time scale vertible. Bioscopic techniques and application. Many type of techniques and application in biophysical, like ultracentrifugation, electrophoresis, size exclusive chromatography, spectroscopy, absorption spectroscopy, and fluorescence spectroscopy, and mass spectrometry, and X-ray crystallography, and nucleic magnetic resonance spectro (NMR), and electron microscopic.
6.
I will explain technique and applications. Ultracentrifugation to separate molecule and different sizes based on the sedimentation principle up to 106 gram. Electrophoresis to separate molecule of different molecule mass size based on the sedimentation principle. Electric field act on the charges molecule gel electrophoresis. uh, Size exclusive chromatography uses tightly packed gel beds and sedimentation based on gravity and sometimes pressure to the trap small molecules, and allow large molecule to pass through the gel faster than small molecules. Spectroscopy. Most of it indicates EM radiation and measuring the intensity, direction, polarization to emit radiation, originally only visual spectrum. 380 to 750 nanometer was used, now also UV, ER, radiation, EM, also electron and mass. Next is absorption spectroscopy, employed as analyzing chemistry to determine the presence of particular substance in small many cases to quantity the amount of the substrate present. Next is fluorescence spectroscopy. Uses a beam of light that excited the electron in molecule of certain compounds and cause them to emit light. Mass spectrometry. Efficient method to include the chemical composition of a small and large molecules. X-ray crystallography to solve the structure of protein and to determine function and residency, interaction with substrates and interaction with other protein or nucleic acid. Last is electron microscopy. Obtained high resolution images of biological and non-biological species. So I am starting now part 2nd of this video. It's amino acid. What is amino acid? All proteins are built up of 20 type of amino acid. Residue of a number of amino acids that are not contained in protein participate in metabolism. Amino acid originate on the earth at the first stage of chemical evolution about 4 into 10 to the power 9 years ago as a result of chemical reaction in the primordial reducing atmosphere and the ocean upon absorption of energy. The simplest amino acids are also encountered in meteorites. Protein contain the residue of a alpha amino acid which has structure. Amino acid are basically building unit of amino acid protein. Um, I will show the simplest amino acid structure. This is a single carbon molecule which is surrounded by amino acid and carboxylic group and hydrogen group, and R for radical group. Where R is radical, a hydrocarbon radical or radical that contain apart from C and H. C for carbon and H for hydrogen. Other atom too as an has been shown by a study of electrical properties of amino acid. Their structure is expressed more exactly by the formula of dipolar ions, also called Zwitterian.